Okay, so what we're going to do today is get into what I consider to be the actual field of genetics. We talked last time about meiosis and how it differed from mitosis. I think you all remember the rule for mitosis was double, then divide, and we end up with two cells that had the same genetic components as the original cell.
In meiosis, you multiply, divide, divide.
Correct. Double, divide, divide, and you end up with 4 daughter cells, and each one of those had half of the information. Again, a reminder, the key there is that it allows for sexual reproduction, which allows for greater variation in terms of the genetics, and that allows for greater survivability across changing habitats. So, we talked about polyploidy, if you remember, and how certain species can have multiple copies of the genome. Humans are diploid, that's what we're used to. Most animals, again, are diploid, 2 copies. 2 complete sets, one from mom, one from dad. If you remember the implications of that, most of the time you see polyploidy is in plants, specifically crops. That is how we have generated a lot of variation. You all remember me talking to you about the differences between the 2 forms of wheat, durum wheat, which is useful for pasta, bread wheat, which has a very different flour and is useful for bread. Durum wheat was a tetraploid. Bread wheat was a hexaploid, 6 copies. Some of you asked what the highest number is that we might come in contact with. There are some species of corn that are actually octoploids, 8 copies. And if you wonder how much variation is possible in corn, just think of all the different types of corn we have. Sweet corn, I'm sure we're familiar with that, right? Flint corn, those are the ones that we get a lot of starch from. Oil corn, I'm sure we've all used corn oil at some point in our cooking. Most of us like popcorn. There are some that are waxy. You get an idea here. But I want to reiterate 2 things. Number one, all of these variations involved multiple complete sets of chromosomes. Multiple complete sets. We didn't have a variation in numbers between sets. If you remember, they also involved even numbers, so that the chromosomes could pair up and separate during meiosis, allowing for sexual reproduction. And again, we had the oddball out. If you remember the example of bananas, triploid. Since they have an odd number of sets, they cannot undergo meiosis, therefore they cannot generate gametes, therefore they cannot reproduce sexually. They're all clones. So what happens when we get a variation in number of individual chromosomes between sets? In other words, we get an extra chromosome. We're missing a chromosome. We've got a couple that are missing. Well, the answer is that is a condition called aneuploidy. Aneuploidy. And I've written it up there for you on the board. That one is not exactly easy to pronounce. By and large, this occurs because you get what's called non-disjunction. Non-disjunction. In meiosis 1, we had the homologs separate, right? Then in meiosis 2, we had the sister chromatids separate. If either of those chromosomal divisions fail, when the spindle pulls the chromosomes to one side or the other, you're going to get one extra pulled to this side and one less pulled to this side. Normally, that condition is fatal. Most of those gametes will either be incapable of fertilizing, or if they do, the resulting embryo will be unable to develop properly. A lot of human miscarriages are probably due to something like this. In general, you need an exact amount of genetic information, no more and no less. There are a few exceptions. In humans, if you remember, we said one complete set is 23 chromosomes, right? Now the chromosomes are numbered from biggest to smallest. So chromosome 1 is the biggest, chromosome 22 is the second to last in size. The sex chromosomes are unique in that they are similar but very different. They are the smallest ones. If you have trisomy in a large chromosome, in humans, it is always fatal. Always. There are no exceptions. If you have an extra copy of one of the smaller chromosomes, it is possible that the embryo may survive and develop.
Is there any side effects if it was smaller?
So something like chromosome 21, for example, that's probably the one example that most people are most familiar with. The condition is called trisomy 21, which means you have 3 copies of chromosome 21 rather than 2. Does anybody know the common name for that? Down syndrome. I think most people are familiar with individuals who have that. They tend to have a delayed development, both physically and mentally. They have a characteristic fuller face, shorter stature, some difficulty with physical coordination. Now that shows you it is survivable. It is also quite possible for someone having trisomy 21 to live a full and normal life. The point I want you to see, though, is that if it is a bigger chromosome, it's not survivable, and generally those particular pregnancies will simply not proceed. Reagan?
So is that like, it ties into like any disability, right?
Not any disability. Keep in mind when we talked about the difference between a mutagen and a teratogen. Remember we said that a teratogen causes a birth defect, but it does not change the DNA.
Okay.
So anything that interferes with that developmental process. Yes, sir.
Just quick, shouldn't the trisomy, shouldn't that be 47 for the total?
Yep. Thanks for catching that.
Yep.
Where it really gets odd is when you have trisomy in the sex chromosomes. The sex chromosomes, as I said, are pretty much unique in the human genome. The X chromosome is a normal chromosome. It has multiple genes on it. It is necessary for function. But the Y chromosome doesn't really do much of anything. It only has one region that is considered essential, and that is called SRY, capital S, capital R, capital Y. For a long time, they didn't know exactly what particular gene on this little branch made it necessary for function, and that's why they called it sexual region of the Y chromosome, the sex determining region. Well, finally, a few years back, they isolated the gene, and the gene is called TDF, testes determining factor. And that little single gene is what determines whether or not an embryo will develop as male or female. If you recall from our previous discussion, what is the default setting for every embryo? Female. So unless that particular gene is expressed at the proper time, and that's the key, then the embryo will develop as female. Is everybody clear on that? And that goes back to something you were asking, Reagan.
Mm-hmm.
When we'll talk about nullosome in a moment, we're going to see what happens if you eliminate that. You just remove the chromosome entirely. It is survivable. Since the sex chromosomes are the smallest, generally, if you've got an extra one or even two, it's survivable. If you're missing one or two, it's survivable. Pretty wild. Yes, sir.
But isn't there rare cases where, like, you can have a male and a female at the same time?
I'm going to get to that. So let's just assume you've got 3 copies of the X chromosome. Your genotype is XXX. That is actually called triple X syndrome. It is trisomy of the X. Not only is it survivable, these individuals develop as females and almost always have normal function, normal fertility, normal phenotype. You don't see any kind of symptom. They can actually have kids. Most of the kids are born normal. It's not something that is really a problem. Most of these do have some kind of consequence, though. For example, there is something called XXY that is known as Klinefelter syndrome. KLINEFELTER. Klinefelter syndrome. So we know that females are XX and males are XY. So what happens when you get trisomy XXY? Well, technically, that person develops as a male because they do have the TDF gene. But they will also have some developmental issues. Compared to the normal range of male phenotypes, they will typically be smaller in stature, more slender. They may suffer from from what's called gynecomastia, which is the development of breast tissue. Typically less muscle development. And lower fertility. There's also an interesting case called XYY, trisomy XYY. And this particular syndrome ended up being responsible for one of the worst scientific studies ever conducted. Individuals who have this genotype will develop as male. Developmentally, in terms of mental capacity, it can range from slower development to normal. Slower to normal. Physically, it can be in the normal range leading up to slightly larger, typically more taller than heavier per se. Back in the 80s, there was a particular researcher in the United Kingdom who hypothesized that individuals who had genotype XYY would be super males and produce more testosterone and would therefore be more likely to engage in violent crimes. So to test his hypothesis, he went to a population where he sampled the individuals in the population and determined their genotype. And he found in this population that the population in question had a higher number of XYY individuals. And furthermore, those same individuals had been convicted of violent crimes. Proved his point, didn't he? Well, the problem was he sampled 4 people with XYY who were guilty of violent crimes by going to the maximum security men's prisons in the United Kingdom and sampling the individuals who had been imprisoned for convictions upon violent crimes. Does everyone see the problem there? You remember when we discussed the scientific method at the very beginning of the semester, we talked about the necessity of good methodology and controls using a blind study or a double-blind study. I don't think anyone has to be a published scientist to see the problems. And almost immediately after it got published, the study came under a lot of fire, rightly so. Because if you were going to check this, you should sample the male population blindly. So that is 3 different examples of trisomy involving the sex chromosomes and 1 involving chromosome 21. There are other forms of it, but I don't want to get hung up on this because I know it's not a genetics class, and you all know how much I love genetics. I don't think we need to be going down that rabbit trail. Yes, sir?
But can it happen to animals as well?
Yes, it can happen to animals as well. Now, one thing to remember is animals don't necessarily follow the same rules as humans. By and large, they do, but in some cases, they don't. For example, butterflies, they don't have the XY sex determination system we have. They have Z and W. And in their case, ZZ is the male. ZW is the female.
Is that the only thing that has that? It's just butterflies?
No, there are all kinds of different things when it comes to this. There are certain organisms that have both sexes on them. Earthworms are a good example of that. There are certain ones that change sex over their lifetime. They'll start as male and transition to female. There are certain bees. Remember we talked about haploid and how that's for gametes? Well, believe it or not, those unfertilized haploid embryos will develop. Those become the males. The queen will be a normal female and she'll have normal eggs, but you've got a diploid queen and haploid males. The males fertilize the queen and then die. You've got some, there are certain species of reptiles that can literally change sex based on the temperature at which their eggs are incubated. Remember we said, you asked the last lecture, I believe it was you that asked this, about how much of this is determined by genetics and how much it's determined by environment. That's a perfect example of where the environment plays a key role. There are others where if you have too many males in the area, some of them can switch to females and vice versa.
What can do that?
Fish, if I remember correctly. It's not one I had researched. I'll have to look it up. It's been a while since I taught genetics. One of the plants that I worked on was what we called gynodioecious, which meant that the plants were either hermaphroditic or female, but there were no males at all. The advantage there, if you're a female, you put all of your energy into the egg. You don't have to worry about making pollen. But if there's not a population of hermaphrodites nearby, you don't get fertilized. The hermaphrodites can self-fertilize, but they don't produce as many seeds or as big seeds as the females do. And you actually had interaction between genes within the genome that could turn it on and off. And people ask me why I like genetics. I'm bringing up humans mainly because in humans, this is the only type of trisomy where you can see that level of variation that's survivable. In the others, it's just one syndrome or another and that's it. Good question though. So, monosomy. Monosomy is when one pair doesn't separate. So you get the pair pulled to one side, and that's going to give you trisomy, but the other one doesn't get anything. Monosomy is almost always fatal. Missing information is not a good thing. In humans, the only type of monosomy that I'm aware of that is survivable is something called Turner syndrome. Turner syndrome. And individuals with Turner syndrome only have a single X chromosome. They are not XX, they are not XY, they are just X0. They literally draw it as a 0. They typically will have some physical developmental issues. Obviously, since there is no TDF gene present, they will develop as female. They tend to be a little bit shorter, a bit stouter. But again, the range can be well within the norm. There might be some developmental delays mentally, or they might just be completely normal. I was actually teaching this not too long ago, and I had a student come up after I gave the lecture, and she said, "It was so cool you were talking about that." And I said, "Really, you enjoyed it?" And she said, "I have it." Would have never known had she not shared it. Well, we talked a little bit more about it. But notice that you cannot have monosomy of a Y. That's because the Y chromosome only has one little chunk of DNA that has any significance. Yes, sir?
So you can actually find out if you have one of those in your body?
Yes, you can. Basically, if you think you would have it, or if there's a reason, you can get tested, and they can do what's called a karyotype, which is where they actually visualize the chromosomes on a slide. Or nowadays, they've gotten to where you can go in, just get your finger pricked, they take a blood sample, and they sequence your DNA. Where they have come within the last 10 or 15 years with DNA sequencing is so over the top that they actually can, basically, you swab your mouth, you send it in, pay them a couple $100, they send you back a report a few weeks later that basically lists every single genetic variation you have in your entire genome and all the possible outcomes. It sounds cool, but the problem is a lot of people don't know how to interpret what that means. We've already talked about that, but that's why we're talking about genetics today. But again, I want you to remember, you cannot be nullosomic for anything, or monosomic for anything except the X. If it's one of the bigger chromosomes, the fact that you only have one copy is going to be fatal. Which brings us to nullosomy. Nullosomy is when you're missing an entire chromosome pair. Instead of 46, you only have 44. That is always 100% fatal. There are no documented syndromes of this in humans, because every one of those 23 chromosomes that you have in a set contains needed information. Everybody clear on that?
So what's it called again?
Nullosomy, it's the third one listed. And then what might be the most bizarre is something called tetrasomy. Tetrasomy. Tetrasomy means you have 4 copies of one chromosome. So instead of one pair, you literally have 2 pairs. And believe it or not, there are a couple of syndromes involving this. You thought triple X syndrome was rare. Imagine the reaction when they discovered quad X syndrome. Females with XXXX genotype. Obviously developed as female, right? For all intents and purposes, since the number is even, they appear to be completely normal in all respects. Fertility, mentally, physically, developmentally. Any questions?
Can they have different than X, like 4 X's? Can it be like?
Almost always, if they were to have a child, it would be with a father who had normal gametes. So it would probably be, they would have daughters that had triple X or sons that had Klinefelter. There have even been documented cases of XXYY males.
From the looks on several faces, I think this has surprised a few people. It's not quite as simple as you thought, is it? Again, though, the sex chromosomes have the advantage of essentially being the smallest in terms of the information that they carry. The more information on a chromosome, the less likely it is to be able to be survived if you start changing it, duplicating it, or something like that. So I just wanted to bring this up to illustrate to you that there's a lot of variation out there. And you brought up something, I believe you were talking about intersexed people, is that correct? People who develop as both male and female. Generally, that is due to a developmental issue, not necessarily a genetic issue. Does everybody understand the difference? Remember, when we talked about mutagens, mutations, we said that we changed the DNA, right? Everybody remembers that? If you remember, the teratogens don't rewrite the DNA, but they interfere with its expression. Remember the children of thalidomide, the flipper babies? We talked about Accutane and why that's not widely prescribed. So when you have an intersex situation, it's very often due to something either derailing one of the developmental pathways, say you're developing as female, and then after the critical point it derails towards the male, or you're developing as male and it derails back towards the female. Everybody see how that goes? In that case, you're going to get somebody who's intersexed who has characteristics of both. There are a couple of other oddities out there. You want me to talk about those? De La Chapelle syndrome. De La Chapelle syndrome. D-E-L-A-C-H-A-P-P-E-L-L-E. De La Chapelle syndrome is when you have an individual who has genotype XX, but phenotypically develops as a male. And what has happened, do you remember when we talked about crossing over in the very first part of meiosis 1? We said the homologs could swap genetic material. Even though the Y chromosome is not homologous to the X, it will pair up with it, and sometimes it can actually perform crossing over. So what you end up with is a gamete that has an X chromosome, but a little bit of DNA translocated from the Y. And since that gene is present, if it's expressed at the proper time, the embryo develops as female. Yes, ma'am?
So, like, say I have a kid and they have male and female parts. How do they decide, like, they have to have surgery? How do they decide which to continue as?
From what I understand now, they don't decide. They generally leave that individual alone until they are old enough to make an informed decision. In the 60s, they would basically make an immediate decision and surgically correct generally towards the female because that's the easier surgery. There was a case in the 60s involving the Reamer twins. identical twin males. And during a botched circumcision, one of the twins had more tissue destroyed than was viable. So they surgically corrected that and raised that particular twin as female. When that particular one at their adolescence, they made the choice to, rather than live as female, but to live as a male. And apparently, despite being raised as a female child, completely and totally rejected the role. And as an adult, having reclaimed the male identity, actually married, had stepchildren, not able to have children of their own, obviously, committed suicide in their early 30s. And that is considered one of the most unethical scientific studies ever conducted. The scientist or doctor who was behind that was far more interested in examining theories of nature versus nurture and how much the environment influences and that sort of thing, rather than actually doing what was best for the child. And as part of the backlash for that, that's one of the reasons why the treatment guidelines basically say, don't do anything. I'm not the one who did it. You don't have to give me that look. Yes, ma'am.
Okay, so just on the topic of like twins, what about, there's people that are like, merged together, I guess, for lack of a better word. Like they're in the same body. Do you know what I mean? They have like two heads.
It's called
like
mosaicism. Mosaicism.
Okay.
What
I didn't know if
happens-
that was genetic or.
In that case, oh boy, y'all are asking the tough ones today. What happens in that case, typically during pregnancy, the woman becomes pregnant with twins, and y'all remember the difference between fraternal and identical, right? Identical come from a single fertilization event, fraternal from two. In the case of fraternal twins, where that happens, essentially, one of the embryos gets absorbed by the other in the womb. I don't know the exact physiology behind it, and frankly, that's well beyond the scope of what we're talking about. But I think you can understand how that would result in an individual who develops essentially with two different genotypes. Is that what you were referring to?
Yeah, like when they're attached and stuff like that,
Yeah.
and they share a body, yeah.
There was actually a case where a woman gave birth to twins, and one of her twins was not related to her because that particular egg came from the other twin. Yeah, there's been some really bizarre scenarios out there. The point is, and this is really the take-home message from all of this, you remember when we talked about the role of the environment and the role of genetics? Do you remember what I said? The genes determine the potential. The environment determines whether or not that potential is realized. And there are a lot of opportunities for errors and mistakes in this. Now, what we've talked about up till this point are large-scale changes. Make sure you've noted that. These are changes that involve whole sets of chromosomes or whole chromosomes. They are the rare kind. None of this stuff is common by any stretch. What's more common are the traditional genetics. And genetics is a field developed in the 1860s. The father of modern genetics was named Gregor Mendel. Gregor Mendel. And Mendel was an interesting fellow. He was a monk who basically lived his entire adult life in a monastery. And every year in the monastery, he grew hundreds of pea plants. Peas like we eat. Green peas. And the reason he did this was because he could grow a lot of them, he could grow them quickly, and he picked traits that had visible variation per generation. Visible variation per generation. Yes, sir.
Didn't he want like mutations for them so they keep growing?
That's what I'm getting to. Things basically like green peas versus yellow peas. Flowers that were purple or flowers that were white. Were the peas perfectly smooth and round, or were they wrinkled and kind of shrunk? The point I want to bring up here is that nobody at the time knew the basis for inheritance. Nobody could say why certain offspring looked like their parents and why certain offspring were different. Remember, DNA wouldn't be discovered for almost another century after Mendel. That's one of the things that make this guy such a genius. He is definitely one of the top 2 or 3 most influential biologists of all time. And Mendel's real gift was that he was observant and he was careful. He recorded everything. And he was the first to come up with something we call an allele. A-L-L-E-L-E. An allele. An allele is simply an alternate form of a gene. An alternate form of a gene. It doesn't mean that it's non-functional, it doesn't mean that it's functional, it just means that that particular gene has different forms. And he furthermore came up with the idea that parents could supply their offspring with alleles that were the same or that were different. And after about 5 or 6 years of doing all of this breeding, he summed up what he referred to as the 3 laws of inheritance. The 3 laws of inheritance. And the first law is what we call the law of segregation. And the law of segregation works because individuals are typically going to be diploid, right? So how many copies of each allele will they have? 2. 1 from their mother, 1 from their father. The point being, these separate during gamete formation. They segregate or separate during gamete formation. So any sperm or egg is only going to have how many alleles?
1.
1. Good job. And what this means is,
traits can essentially skip a generation, can't they? It's why you don't have to have the same trait be visible every generation. And the second one is called the law of independent assortment. The law of independent assortment. And the law of independent assortment says what happens at this trait has no effect on this trait. So in other words, if you have a pea that is green and you cross it with one that is yellow, and the green pea also happens to have purple flowers and the yellow one happens to have white flowers, the color of the flowers has no bearing on the color of the pea. Does everybody see how the traits assort separately? He got lucky. Of the 7 traits he picked, all 7 were on different chromosomes. Remember, we didn't even know what chromosomes were at the time. It turns out that certain traits are what we call linked. And linked traits appear close together on the same chromosome. I always wonder what would have happened if he had picked 6 traits and 3 of them were linked. We might not know the name Gregor Mendel today. And then the final one is probably what most people think of when it comes to genetics, and this is called the law of dominance. The law of dominance. And essentially what this says, there is one allele that is always expressed when present. And there is another allele called the recessive allele that is only expressed or seen when the dominant allele is absent. And Mendel figured all of this out by himself simply by breeding pea plants, counting the numbers of offspring, and repeating and doing other crosses over several years. And after about 5 or 6 years, he wrote all of this up, published it in some very, very obscure journal where about 3 people read it, and it was promptly forgotten for the next 30 years.
Is that at a museum today?
Huh?
Is that book still like around here?
Oh, it's definitely known today. That's why he's known as the father of genetics.
Oh no, I mean like it's still like the book's still.
Yeah,
intact.
you can actually go online and download his data. When I taught genetics, I actually would download Mendel's actual genetic data, the crosses that he performed, the numbers of offspring that he did, and I would assign those as problems. It shows you just how much of a genius the guy was. About the turn of the 20th century, around 1900, several different researchers across 2 or 3 continents started testing similar ideas to what Mendel had. And they wrote letters to each other, they published papers, they critiqued each other, and they started to piece it all together. And then one of them went digging in a very old library in some musty corner and pulled out this old journal, and he happened to read German, and he read Mendel's article, and he said, whoops, guess what, guys? We got scooped 35 years earlier. And that's how we now know Mendel's work today.
That's how many people still have to study him, basically.
People still study him. Every single genetics course I have ever been involved with, there's a chapter on Mendel. and rightly so. Most biology classes talk about him. In fact, this is called Mendelian Genetics. Mendelian Genetics. Yes, ma'am.
I'm just wondering if you can repeat the name of the second law.
The second what?
The second law, I just wondered.
The second law is the law of independent assortment. It basically says that what happens at one trait has no bearing on another trait. In other words, if my plant has purple flowers and green peas, and I cross it with a white flower with yellow peas, the pea color has no influence on the flower color. And the flower color has no influence on the pea color. Yes, ma'am.
So before Mendel and stuff, what did they think was wrong with the people who had Down syndrome?
Demon possession, the sins of the parents. I'm not being snarky. They knew, for instance, that swamps typically cause disease, but they thought it was something in the air. Germ theory didn't come about until about the time Mendel was doing his work. They didn't even know, we talked about the Black Death, for example. They did not know what caused that until the 1890s. The Civil War, this might surprise you. The American Civil War, 1861 to 1865. Does anybody know what caused the most deaths on both sides? Was not combat, it was not gunshots, it was not explosions, it was disease.
Didn't George Washington make his soldiers get vaccinated?
He did, but that was the American Revolution, which was 90 years before that. Good questions though. The simple fact of the matter is that's why Mendel did his work, because people wanted to know, what do you get from your parents? If you remember the last time when we talked about the overall, we talked about heritability, right? So heritability, just to reiterate what we said last time, it's the percentage of a trait that is determined by genetics. It is very, very variable. It's almost impossible to calculate because the environment is so dynamic and changing that it's going to have an effect on the phenotype. It's a great question, by the way. This is one of the reasons why we do science, because we want to understand. We want to know what causes this. So I'm sure that you all did Punnett squares in high school. For those of you who didn't or who may have forgotten, I'm going to show you how to do one. We're going to do a very, very simple cross. We start with the P generation. And let's pretend we're looking at flower color, okay? We're going to say that a true breeding purple strain of flower is crossed with a true breeding white strain. Now, true breeding means as long as you breed them against themselves, they breed true. Does everybody understand what I mean by that? We talked a little bit about inbreeding the last time and how with certain breeds of cattle and dogs, you want that. So it basically means that these guys, both alleles are going to be the same, okay? And we are going to say that purple is dominant to white. And by that I mean the first filial generation, which is just another word, fancy word for saying the offspring. All of the flowers are going to be purple. This was Mendel's first clue as to what was going on. Now remember, if we bred the white flowers against white flowers, we'd always get white flowers, right? So we know there's a genetic component. Purple against purple,
they're always purple. We know there's a genetic component. But every single time he crossed purple with white, he always got purple. And that is why he said that purple is dominant to white. Has everybody got that? So he thought, well, I wonder, are those flowers I got in my cross true breeding? So he decided to breed them against themselves, allow them to self-fertilize. If they're true breeding, all of the next generation should be purple, right? So we represent that basically by drawing it back on itself. And we have the second filial generation, or the grandchildren. Well, he didn't get all purple. What he got was a ratio of 75% purple and 25% white. A ratio of 3 to 1. We skipped a generation, didn't we? Well, here's where his particular genius came together and he showed us what was going on. He said, I'm going to represent the dominant with a capital letter. And we're just going to pick A because A is easy. The true breeding, that means both copies have to be the same, right? We are going to cross that with another true breeding strain, but I'm going to say those are recessive, and I'm going to represent them with a lowercase letter. And from that right there, he figured the whole thing out. The Punnett square came along 30 or 40 years later. Punnett was one of those half dozen or so people who was trying to redo Mendel's work, and he figured out a way to use a graph. So he said, let's put parent number 1 up here on the top. Let's put parent number 2 over here on the side. Now these guys are diploid, so they have 2 alleles each, right? But since those are the same whenever it undergoes meiosis, it doesn't matter if they go this way or if they go that way, each one is going to have the same exact allele, right? So we're going to put a capital A in every box, because this guy would give sperm that had this or sperm that had that. It doesn't matter because they're identical.
Would you put the row A in there as well? Because
one thing at a time. We start with the top, then the side. So what's this one going to be?
AA, the capital.
The capitals, you got it. Those are all possibilities. Does everybody agree? And now we're going to come across, this guy only has the lowercase option, right? So we're going to put a lowercase there, and a lowercase there, a lowercase there, and a lowercase there. Notice that every single one of the offspring have how many different alleles? 2. One is the dominant, and one is the recessive. Does everyone see that? And that is exactly what dominant means. If you have one copy, you get the trait. You may want to write that down. Dominant means if you have one copy of that allele, you exhibit the trait. If you have 2 copies of the allele, are you going to exhibit the trait? Yes, because all I said you need is 1. Yes, ma'am.
All of my siblings have like reddish or brown hair. My sister has blonde. Is that like, do you know what I mean?
I know what you mean.
I'm trying to like.
But notice that we are talking about one trait, one gene, 2 alleles. That's all, right? there are traits that are called polygenic. Poly means many, genic means genes. If you want something that will keep you awake at night, try figuring out a polygenic trait that has 6 genes, each with 6 alleles. And I'm not trying to make light of it. Remember when I said you got lucky?
Mhm.
I wasn't lying, because if a single one of the traits you had picked was polygenic, it would have blown the whole thing out of the water. Sometimes it's better to be lucky than good. Sometimes it's just a case of both. The right person, the right time. And I mean the people that were trying to recreate what he did, they're doing it 35 years later. Multiple minds looking at this, none of them were idiots, and they still are struggling.
So would they still, like, the people 35 years later, would they still have found this later on? if they
Because
didn't?
this is a law, that's why we said laws. You remember what I said that means? It's as close to fact as you can get in science, isn't it? It means every single test we give that hypothesis is supporting it. Every new test we come up with on that hypothesis supports it. Now think about how long ago these laws were discovered, 160 odd years ago. A little bit over a century and a half, and they still hold. And we have tools Mendel could never have dreamed of. This is done 90 years before DNA was discovered. Blows your mind, doesn't it?
I bet if he was alive today, he would be ecstatic about all this new technology. He'd probably mess with it.
Technology can be challenging.
Until it talks to him.
Huh?
I said until it talks to him.
Till it talks to him. I told y'all about my mother in the car, yeah. But you laugh, I still have not adjusted to Windows 11. When it updated on my laptop, I couldn't figure out how to turn it off. It ended up corrupting the hard drive when it shut down, and I had to have IT essentially reinstall it. And even then, I had to go online and look up a video to tell me how to turn off my computer. But the basic principles remain the same. And what Mendel discovered 160 years ago still holds true today. And it allows us to predict. So he decided that these guys were going to be this genotype right here. And now I'm going to show you how to do a Punnett square to figure these guys out. So our new cross, since we are self-fertilizing, is going to be this. Does everybody see how we got that? So pull down a big A here, pull down a big A here, pull down a little A here, pull down a little A here, right? Big A here across, big A here, little A here, little A here. Remember, if I said it's a dominant, if it's present, whether it's one copy or 2 copies, we're going to get purple, aren't we? Everybody see that? So this is going to be purple because both copies are there. What color is that one going to be? Purple. What color is that one going to be?
Purple. White.
And essentially what Mendel did was dozens and dozens of crosses with different traits, but he found out that this pattern held true for every single one. And now it is possible to predict the results of a breeding cross. Think about how valuable that has been in livestock and agriculture. Pretty amazing, isn't it? The problem comes when you start adding multiple traits, because this goes up exponentially. When you have one trait with two options, it's just a simple four box. Any idea what two traits would be? It's not 4, I'll give you a hint, it's 16. You see how quickly it becomes, but even then, there is a way to use algebra, and it wouldn't matter if you had 3 traits, which would be 64 boxes. I could show you how to do it algebraically in under 2 minutes. This has had implications far beyond just crops and livestock. This has impacted medicine. It has impacted the treatment of infectious diseases. It has impacted parenthood. There's an entire field of medicine now known as genetic counseling, and you may want to get that down, genetic counseling, where literally you can go in, have a DNA test done, your prospective mate gets a DNA test done, and the genetic counselor can give you some idea of what possible diseases your child could suffer from. At one point, I was actually thinking about pursuing that career. There are a lot of implications there. Some are good. Cystic fibrosis. Y'all ever heard of that? Cystic fibrosis is a genetically based disease. Essentially, you have 2 possibilities: the normal gene or the mutant form. If you've got the mutant form, it means that your epithelial cells, those are the cells that line the trachea and the lungs, they produce a very thick, heavy mucus. It builds up in the lungs. It causes lots of lung infections and pneumonia. And when I was your age, the lifespan of somebody suffering from CF was about 20 years. They have since come up with better ways to diagnose it early, to treat it, but it still is not a normal lifespan. They've essentially doubled it, but I think most of you would probably prefer to have more than 40 years. So if you know that runs in your family, would you want to know if you could pass it on to your offspring? Well, if you have one copy of the mutant allele, which is recessive, and one copy of the dominant, you can go to a genetic counselor and they can check your mate and say, yep, your mate has one copy of it as well, so 1 out of every 4 of your children are going to have it. And that could lead people to decide not to have kids or to maybe adopt instead. Or it could say, well, you're a carrier, but your mate isn't, so half your kids will be carriers and half of them won't have the allele at all. This has removed a lot of uncertainty for people. Most genetic disorders are inherited recessively. Make sure you write that down. Most genetic disorders are inherited recessively. That means both alleles
have to be recessive. If you have one copy, you are a carrier. If you have just one copy, you are a carrier. So put it all together, and there's some terminology we need to get down. The first term that I want you to get is homozygous. Homozygous means that both alleles are the same. Both alleles are the same. It can be either dominant or it can be recessive. Questions? The second term is heterozygous. That is where the two copies are different. Inbreeding increases homozygosity. So write that down. Inbreeding increases homozygosity. So that increases the likelihood that both copies will be the same. And if most genetic disorders are recessive, that means it's going to increase the likelihood of a genetic disorder being seen. Does everybody understand that? And this is one of the reasons why so many breeds of dog have genetic issues, because they want to breed true, don't they? So most breeds of any kind of livestock or crop are very often extremely inbred. That's good if you have a dominant trait you want to preserve, right? You want to keep it true breeding. But it can be bad if you allow it to continue with a recessive trait that is dangerous. How many of you have heard of the Blue People of Kentucky? This was a family, the Fugate family, in the 19th century and up into the 20th century, that suffered from methemoglobinemia. And no, I'm not going to make you write that down. I always have to look it up just to make sure I pronounce it correctly, because it's such a long thing. And what it was a genetic disorder in hemoglobin formation. Hemoglobin is the red pigment in the blood that carries oxygen. You've seen people who get very cold out of the water, they say they have a kind of blue lips or whitish skin. That is what the Fugate family suffered from. Essentially, that's how they appeared normally. They weren't like Smurf blue or something like that. It was just more of a tint due to this mutant form of hemoglobin. And it was easily treatable. There's actually a blue pigment that they can give them orally that reduces all of the ill effects. But I think anybody who's been in rural Kentucky knows how bad the roads can be even now, right? Or rural Tennessee. Back in the day, as they say, it would be very difficult to travel a long distance. In my grandparents' lifetime, my own grandparents, getting across my home county was a 2-day wagon ride. When my grandmother moved to Scott County, Tennessee from Alaska, because her sister had got married to a serviceman who lived down there. Once they got off the train, they had to wait a week to send a letter down to where he lived, and he could come up with a wagon to get them to take him to the homestead. That was my own grandmother. So I'm just showing you, it's not possible to get a mate from a long distance away. So a lot of times, you would marry cousins, not necessarily close cousins. But this is one example of how that kept a particular recessive allele expressive for over 5 generations. And it looks like it is 3:15, so we're out of time. Thank you all for paying attention, and I hope you found that enlightening. If you have any questions, please feel free to ask. Those of you that have lab, we will meet down in the lab in 15 minutes. So I hope you do. I'm a maladaptive evolutionary biologist. My genes die with me. We'll talk about it. The worksheet that I posted on Parents Event, you can either use the website, I think on the first one, or you can use the slides.
Yeah, in this. So nobody in my family had it before. And so when they were trying to figure out